All right. Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com Inside Scientific and PeptiGrowth webinar titled Synthetic Peptide Growth Factors, Forging the Path Forward for Regenerative Medicine, Cell Therapy, and Cellular Agriculture. I'm Liam Sanyo from the events team here at Scientist.com, and I'm happy to be your host today. We're very fortunate to be joined by Dr. Kosuke Minamihata, Technical Support Manager for Marketing at PeptiGrowth, and Dr. Jess Kuravila, the Sales and Development Manager at Mifi Bioceuticals, who will discuss the application of synthetic peptide growth factors in cell culture models. Hi, Jess and Kosuke. How are you feeling today?
Great. Thank you for organizing today's webinar.
Great, Liam. Thank you so much.
Awesome. Yeah, I'm really looking forward to getting started here. And the floor is yours whenever you're ready.
Thank you, Liam. Dear viewers, welcome to the presentation on Scientist.com platform, where we're trying to introduce PeptiGrowth, a Mitsubishi company. PeptiGrowth is the product of Mitsubishi forethought, looking forward to the landscape of the production of cell therapy and regenerative medicine products that are going to be coming out in the near future. So we would like to really support the true scale-up of these products as everyday cost-effective therapeutics. Mitsubishi Corporation invested a sizable investment into the intellectual property from PeptiDream, a company which uses a proprietary platform for the discovery of synthetic peptides using the platform called as PDPS. So our venture as PeptiGrowth is focused on providing to the market peptides that are of synthetic origin, that are completely xeno-free and chemically defined. Mifi Bioceuticals is a sister company of PeptiGrowth under the Mitsubishi Corporation umbrella, and it's based here in Hackensack, in New Jersey. And we will be the direct contacts for you in North America for these PeptiGrowth products. Our objectives are to support the business development activities of PeptiGrowth, to arrange strategic partnerships with you, the audience, and to provide warehousing of inventory and provide customer support. But last but not the least, the objective is to listen to the customer and relay the customer experience to our teams for development at PeptiGrowth, PeptiDream, and at PeptiStar, as well as Mitsubishi Corporation. So without further ado, let me introduce my colleague, Dr. Kosuke Minamihata. Kosuke-san will present his presentation shortly. But before that, we would like to begin with a short poll for us to help understand the most pivotal growth factors that are required for the success of your protocols and products. So if you can, please choose one of these, and we can have a comparison while Kosuke-san loads up the presentation. Thank you.
Introduction. And thank you everyone for joining our webinar today. My name is Kosuke Minamihata, and I work as a technical support manager for marketing at PeptiGrowth. In this webinar, I and Jess will be explaining about peptides that can substitute growth factors, which are very important in the regenerative medicine, cell therapy, and also cultivated meat industries. So let's get started with the explanation. First, this is a conceptual diagram of what we do. Growth factors required for cell culturing are generally very unstable, and there is an issue of lot-to-lot activity variation. This is particularly problematic in clinical studies that require GMP controls. And furthermore, some growth factors are produced in biotic conditions and may contain animal components, making them unsuitable for clinical use or as additives in cultured meat. The scalability of most growth factors is not high, resulting in extremely high prices. We solve all the problems associated with conventional growth factors by using functional cyclic peptides. Our peptides are in form of cyclic peptides which have non-natural amino acid, making them highly resistant to degradation by proteases. Moreover, since they are just cyclic peptides without any special tertiary structures, they are highly stable even at the high temperatures and do not denature like conventional growth factors do. And they are completely chemically synthesized, eliminating the issues of lot-to-lot activity variation, and they are all animal component free. Also, since we already have a large facility for making peptides, the production scale can be scaled up easily. By using such large facilities, we can reduce the production cost of a peptide and provide them at a reasonable price. In particular, the cultivated meat industry requires large quantity of growth factors at low price, and we believe that our peptides are the only solution that can meet such difficult demand. Our mission is to bring innovation to the cell therapy, regenerative medicine, and cultured meat industries by utilizing these functional peptides.
So we have currently launched 6 products named PG001 to PG006. And these products include an HGF alternative peptide, TGF-beta1 inhibitor peptide, BDNF alternative peptide, a Nogi-like peptide, and also BMP7 selective inhibitor and BMP4 selective inhibitors. And our most recent product is PG005 and PG006 are very unique peptides that we launched in December 2022. And we are continually developing various types of growth factors, alternative peptide, with the goal of launching 3 to 4 products per year. So let me explain how our peptide work as agonist or antagonist by introducing our peptide one by one. First of all, I'd like to explain about the PG001 and HGF alternative peptide, which is a c-Met agonist. So this is an example of agonistic peptide, and in nature, this HGF hepatocyte growth factor binds to its receptor called c-Met and induces the dimerization of c-Met receptors. And this dimerization process is the trigger for the phosphorylation and following subsequent signaling event in the cells. And our PG001 does the same thing as HGF does. Upon binding to the c-Met receptors, PG001 induces the dimerization of c-Met. And our peptides are not mimic of conventional growth factors or the minimized form of growth factors. Instead, we screen peptide that can specifically bind to the target molecules. And in the case of this PG001, the target molecule was c-Met. And we screened cyclic peptide and then connected them using the appropriate lengths of linkers so that those peptides can induce the dimerization of receptors. So what I want to say here is that sequences of peptides are entirely new and original. The graph below shows basic experimental data of c-Met phosphorylation and also the Hubeck proliferation, comparing the HGF and PG001 activities. As you can see, as our PG001 showed comparable activity to conventional HGF in terms of molarity. Usually, the x-axis of such dose-dependent assay is in the unit of nanogram per milliliter. But since our peptides are relatively small compared to the conventional growth factors, and this in case of this PG001, its molecular weight is approximately 1/16 of the HGF. So, for example, this 2 microgram of our peptides is actually equivalent to 32 microgram of HGF. So you may think the price of our peptide is not reasonable by looking at its quantity, but if you compare the price of the equivalent amount of the corresponding growth factors, I believe the price of our peptide is competitive. Here we show further activity evaluation data for PG001, which were obtained by Professor Taniguchi at the University of Tokyo. And he's an expert in regenerative medicine for liver regeneration using the human iPSCs. And he conducted the proliferation assay and also the cloning assay of the iPSC-derived liver cells using either conventional HGF or our peptide PG001. And the results showed that the activity of PG001 and HGF are comparable to each other. And moreover, in the result of this cloning assay, the number of colonies found using PG001 after 10 days of incubation was statistically higher in the case of using HGF. And in fact, this Professor Taniguchi mentioned that he has faced lot-to-lot activity variation issues with conventional growth factors, and he decided to replace it with PG001. The next product is PG002, which is a TGF-beta1 inhibitor peptide. This is an example of an antagonistic peptide. And TGF-beta signaling regulates cell fate, such as cell survival, proliferation, and differentiation. And so TGF-beta1 inhibition is often used in cell differentiation processes. The general approach is to use small kinase inhibitors such as AAD301 and SP525334. And those small molecules enter cells and interact with receptors to inhibit phosphorylations. However, since they're small molecules and lack specificity, they affect other kinds of receptors as well. So this low specificity of small kinase inhibitors sometimes causes problems such as cytotoxicity. And another approach is to use neutralizing antibodies that works outside of cells, and antibodies specifically block activation of TGF-beta1 signal. However, these antibodies are usually made by mammalian cells such as CHO cells, so they are not animal component free, and their price is extremely expensive. So our PG002 can solve these problems associated with small kinase inhibitors and antibodies. And PG002 has no membrane permeability, so they work outside of the cells and interact with TGF-beta1 specifically. And it is chemically synthesized and therefore animal component free, and we can provide it at reasonable price. And this SBE reporter assay showed that our PG002 exhibited comparable inhibitory activity to the antibody. And this PG002 can be used as an alternative for small kinase inhibitors. The third product, PG003, which is a BDNF alternative peptide. And this is another example of an agonistic peptide, and its working mechanism is the same as that of PG001. The target molecule is this TrkB receptor, and PG003 induces the dimerization of TrkB receptors. The left-down figures shows the result of TrkB phosphorylation and also the reporter gene activation assay, and comparing the activities of BDNF and PG003. And as you can see, the activity of the BDNF and our PG003 are comparable each other. And in this reported gene activation assay, our PG003 showed even higher activity than BDNF. And due to the molecular weight differences, the 10 microgram of PG003 is equivalent to 25 microgram of BDNF. This in PG003, we have some application data obtained by third party. And this is the data obtained by Zigzag Bioengineering by using their proprietary technology called nerve organoid. And they are developing the drug screening platform or regenerative medicine for nerve injuries. And Zigzag Bioengineering evaluated the length and the width of neural axon bundles generated from the cellular aggregates by the effect of PG003 or BDNF. And our PG003 induced the generation of the neural axon bundle comparable to the BDNF, and the quantitative results showed that our PG003 actually generated a longer and thicker nerve axon bundles than BDNF. And here's another application data of PG003. We evaluated the behavior of neurite outgrowths from the SHSY5 cells induced by BDNF or PG003. As the dose of BDNF or PG003 increases, the cells started forming dendritic structures, and the quantification analysis of the images shows that PG03 and BDNF are comparable each other. And this data support that our PG03 possesses equivalent activity, not only in the basic signaling activation, but also in higher order phenotypes of neural cells. The next one, PG004, noggin-like peptide. And noggin is a protein that interacts with various kinds of BMP family proteins such as BMP4 and BMP7 and inhibit their signaling. And noggin is often used in development of organoids such as lung, intestine, and kidney. Our PG204 also work as similar to noggin and interacting with BMP4 and BMP7 families and exert inhibitory activity. The left down figures shows the result of inhibition experiment against BMP4 and BMP7 using either noggin or PG004. And as you can see, as our PG004 showed the same level of inhibitory activity as noggin in molar basis. And since the molecular weight of PG004 is actually 1/8 of the noggin, so this 5 microgram of PG004 is equivalent to 40 microgram of recombinant noggin. And closely related to the previous mentioned PG004, we have recently launched 2 new products called PG005 and PG006, which are BMP7 selective inhibitor and BMP4 selective inhibitor peptide, respectively. And these are entirely new functional peptide, and PG005 and 6 allow us to selectively control the signaling of BMP4 and BMP7 families. And the selective control of BMP signaling was not possible with the conventional noggin or small kinase inhibitors. So those PG005 and 6 are very unique functional peptide. Although we are not clear about how to utilize these 2 products, peptides, but we anticipate that they can be useful for detailed analysis and regulation of BMP signaling, as well as for the development of novel cell differentiation processes. So these are the result of the PG005 and 6. PG005 is a BMP7 selective inhibitor, so it showed inhibitory activity comparable to the noggin against BMP7, but it showed non-inhibitory activity against BMP4, even at the 10 times or more higher concentration than the biologically active concentration of noggin. And on the other hand, PG006 is a BMP4 selective inhibitor, and it inhibited the BMP4 at the same level as noggin, but it showed no inhibitory activity against BMP7. And these are our product pipelines. As I mentioned earlier, we have launched 6 products so far, PG001 to PG006. And as upcoming growth factors, which we are expecting to launch in this 2023 or the early of 2024, we have several major growth factors like EGF, basic EGF, and VEGF, and Wnt3a. We are also developing many other peptides that those are listed here, which includes like TGF-beta superfamilies such as activin A and BMP4 and TGF-beta1, and also interleukin series like IL-2 as well. So if there is any growth factors either in or outside of our list which you have great need, we would be happy to co-develop them with you. So please let us know if you have interested in such a co-development program. Lastly, I would like to explain our peptide supply system. And PG peptides are manufactured by our sister company called PeptiStar, located in Sechi City, Osaka, Japan. And PeptiStar is a CDMO company for peptide APIs and originally established to make the peptide APIs developed by our parent company, PeptiDream. As PeptiStar has the GMP compliant facility for API production, our PG peptides can also be made in GMP manner upon demand. So furthermore, PeptiStar has large manufacturing and purification facilities for peptide with a maximum production scale of 10 kg per batch. So by using such huge facilities, we can supply a huge quantity of PG peptides at reasonable price. Here is a summary of today's webinar showing the benefits of PG peptide. Firstly, PG peptides offer an outstanding cost advantage over conventional growth factors. And PG peptides have high stability, which means the media exchange can be reduced. This is a significant advantage as it reduces the need for frequent replacement and can save time and resources. And PG peptides are animal component free, which is an ideal feature for the use in regenerative medicines, cell therapy, and cultivated meat industry. And we can provide a peptide under GMP control upon request, ensuring high quality production standards, and also we ensure the timely delivery of our products. We also offer co-development of alternative peptide to any kinds of growth factors and cytokines. If you're interested, please let us know. Our products have started selling on scientist.com from April 2023, and we would like to take this opportunity to encourage you to access scientist.com and consider purchasing our products. Our PG001 to PG004 are currently available. If you are interested in other PG005 and PG06, please note that they are not available yet on scientist.com. However, please feel free to contact us if you have any inquiries about these products. And here's our contact information. Please feel free to contact us if you have any inquiries. Jason and I are happy to answer any questions you may have during this webinar. And thank you very much for listening.
Fantastic. Well, thanks so much, Kosuke and Jess, for a great presentation. In just a moment, we're going to answer a few questions from the audience here. But Jess, I was seeing that you had a few frequently asked questions to ask Kosuke. Do you want to lead with those?
Yes. Yeah. Thank you, Liam. So from the first polls, I think we have a clear winner. BFGF is by far the most exciting product that everybody is looking out for. So Kosuke-san, I think most commonly everybody would like to know about the stability of these peptides. Can you shed a little bit of light on how stable the peptides are?
Okay, Jason, thank you for your great question. I mean, that's a very important point. Firstly, as for the long-term storage stability, we have experimental data showing that our PG peptides are stable for over at least 1 year at minus 20 degrees Celsius in the form of freeze-dried products. This long-term stability test is still ongoing, so we are expecting to be able to guarantee further longer storage data in the future. And in addition, since our peptides are like cyclic form with some non-natural amino acids, so they're very stable against degradation by proteases in cultural condition. And also, our PG peptides is just a cyclic peptide and do not have any tertiary structures like proteins. So they're very stable even in high temperature conditions. So actual experimented data that we have right now is that our PG peptides remain stable for at least for 1 week at 37 degrees Celsius in serum-free media. And I believe this high stability of PG peptide can potentially reduce the frequency of media exchange. That can really benefit people who do a lot of cell culture.
Oh, well, that's interesting. Thank you for sharing that. I think the end user would benefit from testing it in their own hands, and seeing how they can incorporate it into their own protocols. So the second question that we have that is currently being asked is, what is the intended scale of manufacturing of these PG peptides?
Okay, thank you very much. And I also mentioned in the webinar, but our peptides are synthesized by our sister company called PeptiGrowth, and they do have a lot of huge manufacturing facilities, and the maximum synthesis scale that they have right now is 10 kilograms per batch. So which means we can essentially accommodate any synthesis scale. So although our current product lineup is in the scale, microgram scale, but as RUO, but we are capable of providing products on milligram or gram or even kilogram scales.
That's impressive. Thank you so much, Liam.
Yeah, fantastic. Thanks a lot, Jess and Kosuke. Yeah, answering a few questions from the audience. And as a reminder, if you do have a question that you'd like to ask, you could submit it using the Q&A panel. So a really great question here. Are there any off-target effects of the PeptiGrowth peptides?
Yeah, that's also a very important question. And we also we conducted the like off-target panel assay during our development phase, and we tested against 68 different targets, and we confirmed that our PG peptide do not have any effect on such 68 different targets at even micromolar concentrations. And that means our peptides are really highly specific to the specific target. And additionally, we also conducted the cytotoxicity assay using HEC293 cells. And we confirmed that our PG peptides do not have any cytotoxicity at a concentration of like micromolar range. And this micromolar range is more than like 1,000 times higher than the concentration of the PG peptide that exhibit biological activity. So under the normal usage conditions, we believe that our PG peptides are really specific and also safe.
Fantastic. Yeah, that's great to hear. We have a question here from registration. How does the bioactivity of the synthetic peptides compare to the respective natural peptides' biological activities in health and disease?
That's a very important question as well. We prepared our peptide to have the comparable activity to the conventional growth factors. Not too strong, not too weak. If it is too strong, maybe some like over signaling is might happen. So that's not really good thing. So we are selecting the peptide and we are optimizing peptide to have the comparable activity to the conventional growth factors.
Fantastic. All right, another question here. What solvent should be used to dissolve the peptides? Are they soluble in anything other than DMSO?
Actually, we recommend DMSO to use for the solubilization of our peptide. That doesn't mean that our peptides are not soluble in water or in buffer or in media. We confirm that the solubility of our peptides is at the range of like at least few hundred micromolars in water or in buffer or media. But the problem is the adhesion to the like containers. So the recovery rate is not high if you use the buffer or media to solubilize our peptide. So we just recommend to solubilize peptide at the beginning by using the DMSO. Jason, do you have anything to add? It's okay.
I think it's very common for peptides to be dissolved in a very small amount of DMSO initially. So maybe a drop or two, just so that you have incorporated everything into solution. And then you can bring that up into your basic media like DMEM or PBS, and then incorporate it into the cells. And you always have a control in place where you do that in a with and without peptide. So we can understand the effect of the activity of the peptide in the differentiation protocols. So thank you for sharing that.
Thank you.
Liam, are there any other questions that have popped up?
Yeah, it looks like there's a question here. How long can the peptides get in amino acid residues until it might no longer be useful to produce them compared to the native growth factors?
Yes, can you answer that question for me?
I think. Well, I'm not sure if this is necessarily. It might be kind of a hard question, but is there a certain length that would be no longer useful to produce? If they're a certain length, maybe if they're really short, it could be easy enough to produce them naturally.
Yeah, so in my conversation with Suzuki-san during my recent visit to PeptiDream, he mentioned that the advantage of the PDPS system, which is a rapid in vitro transcription translation system to identify these peptides, you can make the peptide as short as 20 amino acids. That's the optimal length, and that also allows you to scale up the product to the kilogram quantities that you really want to scale up. The objective here is to provide a functional molecule, an agonist molecule that will drive, cell differentiation, rather than make a large recombinant molecule, which is actually difficult to make beyond a few grams. And if you really are trying to push regenerative medicine to as a regular use therapy, I think you will have to have these alternatives to scale up the product. So the advantage, I think, in these small molecules is to, get a viable products available to you at the scale in which you really want to manufacture the end product, which is the cell therapy or the regenerative medicine product.
Yeah, great answer. Thanks a lot. What's the quality control process for sterility of the PG peptides after lyophilization?
We do the filtration by using the 0.22 micrometer of filter to ensure the sterility in the form in the DMSO solution. And also we do then a lot of cell-based activity assay to ensure its activity is okay. And also we do the purity analysis by using the LC-MS analysis, and we ensure the purity is over 95%, and usually it's like 99 or very close to the 100%. But yeah, those are the tests we are doing before we going to bring to the customers. Yeah.
Kosuke-san, there's a viewer who's asked a question about a PG002. They would like to use it for animal studies, and they would like to know if we have any data on how the animals metabolize this product.
Actually, that data we don't have, to be honest. We have only the in vitro assay data, and we do not have any in vivo data. But we are providing our peptide as a RUO product, so basically it's up to you how you're going to use, as long as it is in the researching stage, but you cannot put our peptides into the humans, of course, but for experiment, you can use it to inject into the mouse or do the in vivo studies as well. And we are also really want to know what happens to our peptide in the in vivo conditions, so we like to do that such kind of experiment for sure.
Excellent. Yeah, great answer. There a question here, and Kosuke, I know you mentioned this a little bit in your presentation, that because the peptides are so much smaller than the natural growth factors, you don't need quite as much of them. But is there a difference in activity per molar concentration between the peptides and recombinant growth factors?
The the peptide that we have right now is so far has the same activity in terms of molar as the conventional growth factors, but we are still developing many other kinds of growth factors, and some are very difficult to reach to the equivalent activity. So in the future, maybe we will launch a little bit weaker peptide or in the future, but yeah, we are trying to make it better to reach to the point of the growth factors, conventional growth factors level. So yeah, that's my answer. Do you have anything to add, Justin?
No, I think you've answered the question.
Yeah, thank you.
Excellent. Another one here. What's the degree of residuals in the cell and in the final product?
Well, that's the very important point. Actually, we do not have such data also, but that's very critical point when we gonna use a peptide in the production of cell product, like in regenerative medicine product or also in the cultivated products. But so we believe that our peptides are very do not have the function to enter into the cell. So the residual amount of our peptides will be extremely low, probably below the detection limit when we wash out medium and do the exchange of the medium. So, but still, there is a possibility that our peptides will taken into the cells by the internalization through the interaction with the receptors. So we have to analyze how much of our peptide will actually remains after doing the culturing things. But we're gonna do such experiment to analyze such data, to get such data in the future, and we will provide it in the future.
Yeah,
Mhm.
great point. Another one here. Can we use TGF beta 1 inhibitor peptide, the PG002? Oh, sorry, that was the amount of, that was the question about the
Yeah.
in vivo studies again.
Animals.
Yeah, another one here. Do PG peptides, do they have toxicity?
Mhm. Yeah, we also tested them in cytotoxicity during our development, and we saw no cytotoxicity at the micromolar concentration range. And we haven't conducted, of course, higher concentration range, but we believe that peptides are not cytotoxic at the condition we usually use in the nanomolar concentration range.
Excellent.
Do you have
I'd
any?
like to add a statement here. I think some of the questions that are asked are very specific to individuals and their protocols. And I think the best way to do this is to actually take a sample and test it in your own systems and do a proof of principle. Of course, Kosuke-san did share some data where we saw cell proliferation. And so some of the questions have been answered, but the interactions within your own system are variable. So I think it's prudent that every end user tests the sample, and we are very generous with providing samples for testing. And then run a basic test in your own system, see how it performs, and if it's an advantage to you, then it should support the scale up of the process as well. So that's the key.
Yeah, thanks very much. So another question here: is it okay to assume that the PeptiGrowth peptides and recombinant growth factors have the same mechanism of action?
Yes, we are designed to, our peptides to have the same function as the conventional growth factors, but there are some, growth factors that interact with, several different kinds of receptors. For example, the BDNF one, actually that one interact with the TrkB receptor as a main receptor, but they also BDNF has also the affinity against the P75 receptors. But our peptide PG003 do not have the interaction with the P75, which is not because it was not designed to have such affinity. So strictly speaking, our PG peptide might have a little bit of different biological activity to the conventional growth factors. But for the case of like PG001, CMET is the only receptor for the HGF. So in that case, we are pretty much sure that our PG001 has exactly the same function as the HGF. Yeah, it depends on basically the growth factors.
Yeah, and also I think PG and its collaborators are trying to develop gene expression data. So hopefully in the future we can provide kind of a wider array of the genes that are expressed and the genes that are affected. This is still a pre-marketing stage for us while we launch these peptides, and the peptides are still being used in experiments by collaborators. So as peer review data comes in on these products, I think there will be a wider acceptance on how they function, within various protocols. So as of now, we are still limited with the amount of data that we can share with you currently.
Yeah, great points for both. Another question here about the animals. They're saying that they would like to test it in animals, and they're asking, how do I get a test sample like PG002, and what is the process to get it? So I know, Kosuke, you mentioned scientist.com. You can get it on there, but maybe for those in the audience who aren't on scientist.com, what would be the best way to do this?
Well, yeah, please just contact us to either to Jess or me, directly, so we can arrange sending free samples to you. So please contact us.
Yeah, definitely. I have added the email addresses right onto the slide there, so you can keep that on. All right, excellent. I think in the interest of time, we'll just have one last question here. So given the potential of synthetic peptide growth factors, what are some of the challenges, do you think, that need to be addressed in order to realize their full potential? And where do you think you see the greatest benefits in the future?
Apart from the price, I believe the biggest benefit of using a peptide is the high stability of compared to the conventional growth factors. And there are many companies providing like special media for or media kits for culturing specific type of cells, typically like iPSCs or ES or some neuron or liver cells. And those specialized products are very easy to use and so accepted by the many researchers and people are buying those. And however, growth factors, conventional growth factors are very unstable. So in those in a specialized media, those growth factors are usually supplied as a completely separated supplement, and they have to add the growth factors just before the use. And we are also, as PeptiGrowth, we are also seeking the possibility to develop such specialized media using our peptide. And since our PG peptides are very stable, and we believe that we can put our peptide into the media and then we can sell it. So we can make the media that is completely ready to use. And I believe that that makes the all many processes of cell culturing much more easy, and we can benefit many people in the cell culturing industries, I think. And that will also expand the use of PG peptide in the future. And however, the biggest challenge to achieve such activities is to increase the number of the product lineup. So far, we have only 6 products, and those are, to be honest, a kind of niche, ones, and not many people are using. Like in order to make such media systems using a peptide, we have to increase the number of the lineups, and we have to have the like EGF or basic FGF, the major ones as well, and also we need to develop like TGF beta superfamilies like BMP4 and TGF beta 1, and also interleukin series. But the challenge is that those growth factors we haven't developed so far has more complicated, receptor binding, behaviors like trimers or tetramers. So the PG peptide for those, TGF beta or interleukin series needs to be probably trimer or tetramer. So we have to accumulate, know-how of how we're going to design the whole linker region or the peptide things. So that would be the kind of challenges for us. But by doing so, we can ideally replace all the growth factors in the market regarding this regenerative medicine cell therapy to cultivate the meat industry by using peptide. That's kind of our future goal.
Yeah, really nice answer. Great points. Jess, what do you think?
I think I kind of agree with Kosuke-san. The challenges are in front of us, and I think the PDPS system is well equipped to actually provide very novel solutions. And as a proof of principle, we've provided 6 of them. But I think going forward, I think the audience can expect that we will try to surmount some of these challenges that we are facing in the market as well. So keep an eye out for us. We will definitely be pushing a lot of literature out, and as well as providing announcements when our products come out on BioInformant and other platforms, and also here on scientist.com. We are hoping to have at least a few more seminars or webinars of this nature very soon as well as the products come out and as we gather more data for it as well. So, I think for us, it's an exciting start here on scientist.com, and we look forward to supplying you with innovative products.
Yeah, definitely. Thanks so much. Well, yeah, thanks again so much for the great insights, Jess and Kosuke. It's really been a pleasure having you on with us.
Thank you.
Thank you, Lee.
Yeah, a big thanks also, of course, to the audience for participating. And in closing, we hope you enjoyed this scientist.com, Inside Scientific, and PeptiGrowth webinar, and we look forward to having you on with us again soon. Have a great day, everyone.
Thank you. Bye.